Okay, so in many of your earlier courses, Intro Cell and Molecular Biology, many of the times they sat there and just said, hey, look, calcium is released, cell does something, and then they leave it blank. They don't tell you what the cell does, or what the cell can do because of this efflux, this introduction of a high concentration of calcium into the cytoplasm. Well, that's what we're going to talk about right now. One of the things that calcium can do is it can trigger certain transcription factors. One of those transcription factors is known as NFAT, NFAT. And here's how this works. Gated ion channel, closed, calcium, high concentrations either outside the cell or this is the ER, so your internal, so whatever. 3 players here. Calmodulin, which we talked about in a previous lecture. Calcineurin, which is going to be one of those effector proteins, and then NFAT, our inactivated transcription factor. Something triggers this gated ion channel to open. Okay, doesn't always have to be IP3, there can be other things. Calcium floods in. Now, as we talked about before, calmodulin in its normal native state is not interacting with anything until calcium binds to it, puts it into a certain shape so that an effector protein here, calcineurin, will now interact with it. Calcineurin bound to calmodulin now recruits in NFAT. It dephosphorylates it. Dephosphorylated NFAT is now actually active. Turns out NFAT is one of those proteins that when phosphorylated, it's inactive. When it's dephosphorylated, it becomes active. And what active NFAT does, it traffics to the nucleus, and it has a certain DNA nucleotide sequences that it will bind to, and it will act as a transcription factor, promoting expression of certain genes. So, calmodulin, calcineurin. Calmodulin doesn't do anything until it binds calcium. Calcineurin doesn't do anything until it binds to calmodulin that's in the correct shape. To be in the correct shape, calmodulin has to be bound to calcium. So calcium bound to calmodulin is now able to interact and recruit calcineurin. These two together can dephosphorylate NFAT. NFAT will then traffic to the nucleus, as I said, bind to certain promoter regions, initiating transcription of a specific subsets of genes. What about that RAS we talked about before? RAS, RAVE, remember all those? Well, they also can trigger transcription of certain genes. RAS, GRP, remember there's going to be certain proteins that are going to interact with RAS, going to bind it, open it up so that it now can become active. Okay, so now you have RAS GRP bound to diacylglycerol. Remember, you cut off take PIP2, cut off the IP3 and release the PIP3, leaving diacylglycerol in the lipid bilayer. Well, that's going to act as a platform for RAS GRP to bind to. It's going to bind there. It's now stable. It's now going to be in the correct orientation and everything to interact with RAS. It's going to take inactive RAS, activate it. Active RAS is then going to recruit in scaffold proteins. One of the scaffold proteins is going to be KSR. KSR is going to then bring in RAF, MEK, ERK. And they're now all straight in a line. RAS activates RAF, RAF activates MEK, MEK activates ERK. ERK will then go and phosphorylate and activate certain transcription factors. Boom. Again, we see certain genes turned on, turned off. And if you think back, diacylglycerol, the only reason why it's going to be there is because, as I said, PIP2 is going to be cleaved. It's only going to be cleaved in the presence of activated phospholipase C gamma or theta, depending upon which cells. And PLC only gets activated when ZAP70 has activated those scaffold proteins, and ZAP70 is only going to be activated in the presence of phosphorylated ITAMs. Phosphorylated ITAMs are only going to be in the presence of active form of LIC. LIC is only going to be active when CD4 or CD8 is interacting stably with MHC. Oh. Again, you look here, ERK. ERK has just got activated. IMEC, as we just talked about, and then traffics to, and here's an example, MEC, ERK goes and interacts, phosphorylates ELK. ELK goes and then binds at what they call an SRE. SRE is a signal response element. This is going to be upstream of a gene. So ELK1 is going to bind, and it's going to push through. Or ultimately, you can have the MAP kinase pathway known as SAP-JNK. JNK is going to be activated, phosphorylated. It's going to go to the nucleus. It's going to activate Jun. Jun is going to bind phos, forming AP1, which is also going to bind to some of those SRE elements, activating transcription. Maybe the cell, instead of the PKC gamma, maybe it's going to be PKC theta. It's going to go through and you're going to see, or the PLC gamma is going to go and activate PKC, protein kinase C theta. Again, PKC, protein kinase C, is bound to diacylglycerol. It is held in place. Different protein known as KARMA1. KARMA1 is going to come through. It's going to interact, and it's going to start recruiting in MALT1 BCL10. All of these are now going to line up and are going to start forming a large complex, which is going to recruit in TRAF. TRAF becomes ubiquinated, releasing TAC1. TAC1 is then going to go ubiquinate also proteins and a complex known as IKK. IKK is going to go and is going to break IKB from NF kappa B. I kappa B from NF kappa B. NF kappa B is actually a transcription factor. It wants to go and it wants to bind to certain DNA sequences to promote transcription. The thing is, in its native form, it's bound to I kappa B. I kappa B interfering kappa B can only be removed if it is ubiquinated. Well, to be ubiquinated, you have to have TAC1. TAC1 then goes and activates IKK. IKK then goes and ubiquinates I kappa B, which causes it to be degraded, releasing NF kappa B as a active form, which will then traffic to the nucleus, bind to a DNA, and transcription activated.